recently moved to the Scottish Royal Family's Preferred Institution at St Andrews. He's speaking on multiscale mathematical modelling of cancer growth and spread.
Thank you, John. Yes, I've just recently moved to St Andrews all the way from Dundee, which is a short distance but a different world. And but the Royal Family have left long ago, independently of my move. So this morning, as John says, I'll be talking about some models of cancer growth that we've been working on recently, and looking at multi-scale modelling. And thanks to the organisers for the invitation. It's always nice to come down here to the Isaac Newton Institute. So the overview of the talk, just a little background biology of cancer. I'm sure you're all aware of what it is and what it does, but just to go into some details of some of the key aspects of cancer growth and spread. And then the multi-scale nature of the modelling is going to try and link up different key scales in cancer. And of course, these scales aren't important just for cancer growth. They're important for all really tissue level phenomena, starting inside a cell, going to a single cell or a few cells interacting together, and then going up to the tissue scale. And so specifically for the cancer modelling, I'll be looking at some recent work on gene regulatory networks inside cells. In terms of the cell scale modelling, looking at models of cell migration for metastatic spread, and I should have removed that last night. I won't be looking at angiogenesis today. And then at the tissue scale level, building on from the cell level, some models of cancer invasion. So if we look at a single cell, because cancer is essentially a disease of the cell, and if you look at the differences between a cancerous cell and a normal cell, so beginning with some genetic mutation which will have arisen for whatever reason, and then leading on through other mutations, if you get to the point of being a cancer cell, there are a lot of differences between a cancer cell and a normal cell. Both inside the cell, in terms of genetic mutations, that has an impact on the cell biomechanical properties. The cytoskeleton of the cancer cell is different, and then that has impact on cell-cell adhesion, cell matrix adhesion, and cell migration at the cell level, and then at the larger level. And then also in terms of what the cells do and how they function, they secrete lots of different types of enzymes, chemicals, etc. And in two main classes. One set of enzymes interacts with the components of the matrix to degrade the matrix, and that is a key aspect of invasion. Another set of factors has the ability to induce new blood vessels. That's connected with angiogenesis. And then there's also the ability of cancer cells to impair the response of the immune system. Modelling the immune response to cancer is a whole interesting area of itself. I won't touch on that at all today. But again, from a cancer's point of view, they've evolved mechanisms to try and fool the body's immune system and stop a good immune response. And all these differences were very nicely summarized in a now famous paper about 15 years ago by Hanahan and Weinberg in the Hallmarks of Cancer, and then they updated it in 2011. Obviously Star Trek fans, so they updated their hallmarks to a few extra hallmarks, but the original hallmarks that they identified as differentiating cancer from normal cells and tissues were the following. There were, and 4 of these really are to do with the enzymes and the chemicals that cancer cells secrete. So we have the ability of cancer cells to keep proliferating, and that's due to some autocrine signaling that cancer cells secrete. They can avoid the body's response to try and stop cancers proliferating, so they evade growth suppressors from the body through various mutations, one of which is in a key gene and protein called P53. They can avoid cell death, natural cell death, apoptosis. P53 is mutated in many cancers, and that allows the cancer cells essentially to become immortal, and they replicate endlessly. So these four are to do with chemicals that the cancer cells secrete, and then these other two, angiogenesis and invasion, are more cell tissue level phenomena. If cancers simply did all this stuff and secreted chemicals and grew into little masses of cells, I'm sure far fewer people would die. It's the ability to get a blood vessel network, which then allows the cancer cells to get into the main blood system, circulate, and that's also connected with active invasion of the tissue. Blood vessels, if they are not actively induced to connect to the tumour, the cancer cells can invade the local tissue and then can encounter vessels in the normal healthy tissue, and of course interacting with vessels, not just angiogenesis but the lymph system as well. The cancer cells can enter into the main circulatory system, be carried about the body, and before you know it, you've got secondary tumours as well as primary tumours. So that is the real complication with cancer. The secondary spread to distant sites in the body makes it almost impossible to treat at that stage. So it's a phenomenon on different levels, spatial and temporal. It's a multiscale problem. This is a section of a breast tissue. The white mass in the center is a breast cancer, carcinoma. This is from some colleagues at 9 Wells Hospital in Dundee. So that's what breast surgeons study and try and operate on and excise if you have breast cancer. And if you focus on a bit more closely at the sort of invasive edge of this tissue, you can see that there's interactions between the cancer cells, which are here stained in purple, and the normal tissue that's in white, the stroma, which is in white. So there's a lot of heterogeneous interactions between the cancer cells and the external tissue. And then if you look inside the cell, that in itself is another complicated little world. So here's a nice stained picture of some cells with a nucleus and microfibrils in the cytoplasm, and these are important for transporting proteins throughout the cell and mRNA in and out the nucleus, and we'll touch on that in the first types of modelling. So just as a little summary, to put that in perspective, here's a nice little movie. This is just a movie one of my PhD students found some years ago on the web, but it nicely summarises cancer. So there's some mutations going to arise in a cell. This is a single cancer cell. That then starts to replicate and proliferate at a faster rate than the normal cells. So you get a little mass of cancer cells growing, forming into little solid mass. This then grows to some steady state of a few millimeters, at which point it becomes starved of nutrients and essentially stops. And then that allows the hypoxic cells inside the tumour to secrete angiogenic factors, and there's the growth of blood vessels into the tumour. So that's the angiogenic process. That brings oxygen to the tumour, and so it expands even further. So you're now getting a large mass of tumour cells. And then you'll see here a little green tumour cell. It's got into the blood system that is induced. And so in there, it is carried into the main blood system. It circulates in the body, and then at some distant site, it extravasates, as it's called, into some other tissue in another part of the body and sets up a secondary tumour. So it's a complicated disease, complicated on many levels, and so I'll now show you some of our work that we've been doing in the last 4 or 5 years. So the first scale, the first level we're going to look at is the intracellular scale. This is a schematic diagram from the original paper of Hanahan and Weinberg, trying to show some of the key pathways that are involved inside cancer cells. Now, in addition to these I mean, this is, you're probably aware of signal transduction pathways. Various proteins, various molecules get phosphorylated, ubiquitinated, all sorts of atids, and you get kind of chain reactions until something interesting happens and the cell does something that it hasn't done before, sort of. And these signal transduction pathways control key processes at the cell level, such as proliferation, cell death, cell migration, and also cell adhesion. For example, one thing I'll talk about later in the talk is the cell-cell adhesion, and that's controlled by two proteins called E-cadherin and beta-catenin. So they are involved in a pathway here. Here's P53. That's involved in cell death, programmed cell death, apoptosis, and that's linked to another protein called MDM2, and all sorts of other pathways are involved in other aspects of the important aspects of cells. So up until recently, when people have been modeling these signal transduction pathways, the great temptation has been to just consider the cell as a well-mixed reactor and write down some ordinary differential equations of molecule A being phosphorylated into molecule B, or molecule B doing something to be into molecule C, and just have a list of ODEs until some level is reached in your concentration of a molecule that does something, and then a cell is activated. And I think that's okay for certain types of pathways, but the pathways the pathway I was mainly interested in was P53, and that's connected to another protein, MDM2. And you can see here some of these little black lines. Some of them have an arrow on the end. That means there's an activation process going on. Others have a little a vertical line, which means there's a negative process going on. So MDM2, there's a little line here, that means it downregulates P53, so it stops P53 being produced. So that's called a negative feedback loop, or a negative feedback system. And that's associated with molecules called transcription factors. And transcription factors are molecules which interact with the genes, some genes, which of course then interact, which plays about with the levels of mRNA, which then messes up with the levels of protein. So transcription factors are important in regulating the levels of protein in a cell. And here's a system called NF-kappa B. Don't worry what it does. Just focus. These are single cells that have been stained with red fluorescent protein. And what you can see here is, I'll just show you that again. The nuclei in these cells sometimes go dark, and then they sometimes go red. Okay, and that's because the NF-kappa B system, there are lots of proteins involved. That's because what's happening is that the protein is going into the nucleus, binding to some gene site there, and downregulating the production of mRNA, which then downregulates the production of protein. And this is happening because the mRNAs and the proteins are going in and out of the nucleus and in and out of the cytoplasm. So I saw this movie about 5 or 6 years ago at a talk, and as soon as I saw the nucleus and the cytoplasm kind of lighting up and going dark, it really struck me that it was a spatial process. And really trying to write down some ODEs to describe this system, to me, wasn't really going to capture the essence of the system. So what we decided to do is look into these transcription factor systems in a bit more detail with an explicitly spatial model, a PDE model, make a little mathematical cell with a nucleus and a cytoplasm, and just move proteins and mRNAs in and out of the appropriate compartments. So these are the systems that we're looking at. These are negative feedback systems. This is a generic feedback loop. So, and you can argue intuitively and get to see the kind of dynamics that are there. If you have some species X, which upregulates Y or creates more Y, and then Y either directly or through other interactions or indirectly then downregulates X, what's happening is that X concentration increases, but as the X increases the concentration of Y, so the concentration of Y rises, but then Y downregulates X and X decreases. As X decreases, that makes less of Y, so the Y concentration starts to decrease. So intuitively, you can see that these systems oscillate. And that's exactly what happens. If you look at these feedback systems, these protein systems, you get oscillations in the concentration levels of the proteins. If you go and plot the concentration of NF kappa B, it oscillates with some period of a few hours or whatever. And people have known that, and they've tried to model it. And if you've got enough ODEs and enough nonlinearity, you'll probably get a Hopf bifurcation and an oscillation. But there's a very simple system right at the beginning called HES1. HES1 is its own feedback system. It's simply the protein and the mRNA. So all that's happening is that protein is produced at some rate alpha, and there's a bit of decay. The mRNA then produces the protein at some other rate, and the protein decays. And then the protein goes back and downregulates the mRNA. So people have known this for many years. This system was first proposed by Brian Goodwin in the mid-60s, and you can choose your favorite feedback function, normally some kind of Hill function, 1 over 1 plus p to the n or something like that. That's fair enough. But in fact, if you look at that with a little bit more mathematics, you can choose any feedback function you want, and you cannot get oscillations in this system. But we know from the data that there are oscillations there. So what people did was they just started sticking delays in as wherever they could, arguing that it takes a little time to make proteins or to move things around. And of course, if you stick in a big enough delay, you'll get oscillations. So for a long time, these feedback systems were modeled with discrete delays in systems of ordinary differential equations, but I still felt that was somehow putting in the answer before you'd even asked the question. So what we did a few years ago is, as I say, to make a little mathematical cell with a nucleus and a cytoplasm, and simply put the molecules in the correct compartments and allow them to move about, initially just by diffusion. So you make mRNA in the nucleus, that diffuses, goes across the nuclear membrane, goes into the cytoplasm where it makes protein. The protein then diffuses and decays, goes back into the nucleus, and downregulates the mRNA. And if you write down the system, you've basically got those ODEs, except in the correct compartments and with a bit of diffusion. So here's our mRNA in the nucleus. It's diffusing and it's decaying and it's being produced, and then it's being downregulated in the nucleus by the protein when the protein's in the nucleus. And then when it gets into the cytoplasm, it just diffuses and decays. And then similarly for the protein, it's produced in the cytoplasm and it diffuses and decays. And then when it gets into the nucleus, it's just diffusing and decaying, and then it's downregulating the mRNA. And if you do this in the cell, you put zero flux boundary conditions on the outer. Yeah.
One question. This now assumes just plain diffusion in a homogeneous medium. Now, through the nuclear pore complex with active transport, media inhomogeneous, is the result sensitive to these complications, or is it just some modification of some number?
No, that's a very good question. So everything you've said is very true, and we knew that when we started out, but because the previous models had been ODEs with no spatial aspects at all, we thought this was the first step. So yeah. Now, subsequent, I won't go into this in too much detail in this talk, but subsequent to this very basic model, we have put in, well, okay, let me just say that across the nuclear membrane, we just assume continuity conditions, so continuity of concentration, continuity of flux, so very easy. But we then subsequently put in a finite sized membrane and had a diffusion coefficient across the membrane, so we could slow molecules down, try to model the fact that they get through the nuclear pores with or without some difficulty. We also put into a later model convective transport in the cytoplasm, trying to model the action of the microfibrils, because the proteins bind to the microfibrils, and then you get motor proteins binding to them, and then they push them to the, yeah. So we've done all that subsequently, and clearly inside the cell, it's not just a nice homogeneous medium. There's all sorts of other things messing things up, so this should probably be some nonlinear diffusion accounting for various things in spatial parts of the, sure. So you can make this as complicated as you want, yeah. But I don't think it'll change the underlying dynamics, which I'm await to show. But that's a very good point. So anyway, so if you start with the simplest thing, which is just diffusion, which is a nice step forward from discrete delays and ODEs, then you come up with a system like this. And within half an hour of coding this up, the first time we did this, we very nicely got some oscillations. So no tricks, no delays, everything oscillated, and with kind of changing the parameters, we got the oscillations to be roughly 2 hours, about 120, 130 minutes, which is the oscillation period of Hess 1. So that was a nice result, and so we pushed that further. In subsequent work, we've added in convection and looked at the influence of the size of the nuclear membrane, the width of the nuclear membrane, and other things. But, yeah.
What's control the period? What's the primary control?
So you can change the period by varying the diffusion coefficient, the production rate, or the decay rate. So three main ways to do that. This is obviously the simplest feedback system you get. I mean, NF kappa B, you may be looking at 27 equations. So people know there are at least 27 key interactions, so there are about 27 equations you have to write down. Obviously then you're into tens of scores of parameters which change any number, you'll probably get a change period. Having said that, some of the molecules in the NF kappa B system are known to be far more important than others. Some are just kind of passive vehicles moving you on to the next stage. So you can normally focus in on a few, a smaller number of key parameters. But in this system, the diffusion, the production, and the decay are, mathematically at least, you can change them. That will affect the period. Of course, biologically, you might expect the decay of a molecule to be kind of fixed. You know, you wouldn't expect that to vary unless you did something drastic to the cells. You probably would also think that under, you know, normal circumstances, the production rate doesn't vary that much, but the diffusion could vary from cell to cell depending on what's there in nuclear membranes and things. So I would say diffusion is probably the key parameter here, but the others also influence the period. Yeah, and in the papers that we published here, we looked into varying parameters from a baseline configuration, increasing and decreasing, and showing what effect that had on both the amplitude of the oscillations and the period. So that's in the work that we've published recently, if you're interested. But so in doing that and changing the parameters, we could see that didn't affect the oscillations. And in fact, if we look carefully at the diffusion coefficient, we saw that if we increased the diffusion coefficient and made it too large, we lost the oscillations. And if we decreased the diffusion coefficient and made it too small, we also lost the oscillations. So we thought that somehow that was like a Hopf bifurcation parameter, and indeed, you know, you do it numerically, you get the kind of classical Hopf bifurcation picture. But we felt that we could do a bit more, so we simplified the model, and we've now made a little one-dimensional mathematical cell with a nucleus and a cytoplasm, so a blue nucleus and a green cytoplasm, and a little gene site here located at some position x, m. So if you do that, you can write down a one-dimensional model. These are mRNA, it's diffusing, it's being produced at the gene site, so that's localized by a smooth delta function, and you've got your feedback function here from the protein, and it's decaying, and the protein is diffusing, it's being produced in the cytoplasm. G of x is a heaviside function, so it's only on in the cytoplasm, and it's decaying. And if you study this numerically, you get oscillations, and it does everything we expected, we hoped it would do. So the simplified model seems to have the correct dynamics. And if you know a very smart young applied analyst, Dr. Maria Potashnik, just recently we managed to show, largely Maria managed to show, that the diffusion coefficient does indeed act as a Hopf bifurcation parameter. So the oscillations are caused by diffusion. So it's the spatial movement that is causing the oscillations, and we've shown that rigorously in this recent paper. So that seems to me to say that if you do model these systems, these feedback systems with ODEs, it's basically the wrong technique to use with delay ODEs, and that's because with delay ODEs you cannot capture diffusion, and here we've shown that it's diffusion that's causing the oscillations. Now, of course, you will get, if you have delay equations and big enough delays, you will get oscillations, and if you change your delay parameter, you can match your output of your model to the data, but I think you're getting the right answer for the wrong reason. So I really think for these feedback systems, you should be using PDE models.
Mark, in the absence of diffusion,
Yep.
your ODE system,
Yep.
what does it, how does it behave?
The ODE system does nothing.
With the delta, now you've modified it, so it's substantially different from the one proposed much earlier.
Okay, again, here, if you have, yeah, that's part of the way we solved it. If you have very, very small diffusion, you essentially localize the production of your mRNA spatially. So it doesn't diffuse far enough to activate protein. So you get mRNA produced here, but it doesn't then get into the cytoplasm to activate the protein.
Right.
So the system has two steady states. For small diffusion, you localize the mRNA here, and because it doesn't get into the cytoplasm, you have no protein if you start off with zero initial conditions. At the other extreme, if you consider very, very large diffusion, so very, very large diffusion means that, well, you can divide by d, and well, you just do the algebra. It's easy. What happens is you get essentially two almost flat steady states, because your first order correction to this equation is d2m dx squared is 0, d2p dx squared is 0 for large d, and with zero flux boundary conditions, you know that solution is to be a constant. So you've essentially got two constant solutions for the mRNA and the protein, and nothing else happens.
I wonder about the dimensionality, because if you look at diffusion-controlled reaction in different dimensions, you find that in 3D you will get a constant reaction rate with a short transient in the beginning. In a one-dimensional system, you have a reaction rate that decays like 1 over square root of the time, and all molecules will react in 1D, and in 3D there is only a finite fraction that will react. So I would expect if you increase the size of your cell more and more and more, you would find different behavior in the 1D and the 3D model. Is that true, or did I miss something important?
So we've done simulations in 1, 2, and 3D, and we get oscillations in all three systems, on all three dimensions, everything. I think you might have to change the diffusion coefficients a little, but you don't have to, you don't have to say have three or four different orders of magnitude or anything. It's a small change, an acceptable change, and in fact, the way we got the system to work is we non-dimensionalized the system. Well, first of all, we went into the literature and thought we'll just look up the diffusion coefficient of Hess 1, and very few biologists seem to be interested in doing that, and even fewer have even. I know there's things nowadays like flip, flap, and fret, and all these things, but even then, few biologists seem to be interested in measuring the diffusion of molecules in a cell. So what we did was we non-dimensionalized the system, we ran the simulations, and we got oscillations, and then we reverse engineered and worked out the dimensional value of the diffusion coefficient, and thankfully it was in a range that was acceptable for molecules. It wasn't 1 kilometer squared per second or anything stupid like that, but which would have made our results mathematically nice but biologically irrelevant. But so you can do it in 1, 2, and 3 dimensions with appropriate diffusion coefficients, and it's all okay. But that's a good point. I'll wait to get to that in the next part. Yeah.
But I guess once you've gone this far, why not simply go to a compartmental model and add a ODE for transport, which gives you a much faster solution?
Sure.
You can certainly reduce these equations formally to get the transport coefficients. so that you'll get something that maps reasonably well.
So, with the P53 system, people have done that.
Sure.
But if you make a sensible assumption about just the nucleus and the cytoplasm and some P53 in the nucleus goes to P53 in the cytoplasm, as you would with say compartmental models and ODEs or metapopulation models, you still don't get oscillations. You then have to make the ODEs more complicated and evoke other reactions that are, in a secondary level. The guy in Paris, Jean Clérambault, has done that. They had to go to something like, so if you have P53 MDM2, the basic system is about 4 ODEs. They had to go to maybe 9 ODEs with some other ubiquitination thrown in just to then make the system oscillate. Whereas if you just take the spatial model, it works very nicely. But yeah, and so the but anyway, getting to a compartmental model. This is sort of a compartmental model, but okay. What we also thought for these results, although they're nice and we got oscillations and the right period and etc. If you look at the real data, you find that the actual numbers of the proteins in the mRNA are actually quite low. So any continuum model is probably a big assumption too far. So you really want to be trying to model the individual molecular interactions, so you're looking at some Gillespie type of model. So we did that. We took a spatial Gillespie model, so we took a 3-dimensional cell with a nucleus and a cytoplasm and a gene site in the nucleus, and we modeled the HES1 system with a spatial Gillespie model. So we have our gene site in red, so all the colors correspond to the locations in the cell. So now with a stochastic model, you don't need Hill functions and all that. You just have your gene site and you have the protein binding to the gene site. So that, and P now stands for the promoter region in the gene. So PF is the free promoter region. That's when it's producing mRNA normally at the rate alpha M, making mRNA, making the protein, which then goes back and binds to the promoter region and shuts it down. So that then reduces your mRNA production rate by a factor of 30 or something. So that's your feedback system there, and you do that in a spatial way with stochastic Gillespie. So you just treat diffusion as a reaction rate. So here's our little spatial stochastic scheme. So the protein binds to the free promoter at a reversible reaction and shuts it down or makes it free. When it's free, you have normal production of mRNA. When it's closed, you have reduced production of mRNA. mRNA makes the protein, or it decays, the protein decays, and then the proteins and the mRNAs all diffuse. And if you do that, okay, sorry, let's see this. So this is data from a group in Japan. They were interested in HES1 levels because HES1 is in mice. Embryonic stem cells is implicated in differentiation. So they were trying to work out levels of HES1 acting as a differentiation switch. And these are their data. And this is the levels of protein in an individual embryonic stem cell from a mouse embryo. So you can see it does oscillate, but in a kind of stochastic way. And the levels of protein numbers are a few hundred. The levels of mRNA molecules are in, say, between 20 and 50. And here's our simulation from the spatial Gillespie model. And so you can see, at least in the eyeball norm, hopefully, that these are very similar, not just qualitatively, but also quantitatively. We're getting the right protein numbers. And here's the mRNA at the bottom, just a few tens of molecules here. And if you do a proper analysis with some kind of time series analysis of the data, you can show that there's an underlying period and there's stochasticity as well, and so that does map very nicely onto the real data. Okay, and then recent work, we've looked at P53 because that's implicated in cancer. So here's P53 and MDM2. I'll just quickly go through this because all the work's been laid in the HES1 system. So P53 and MDM2 oscillate in cells. This is from some work of Gilad Lahav and Harvard. So here's time series of their data. So these are proteins, P53 and MDM2 levels in a single cell, oscillating over time. And again, if you put that system into the spatial stochastic model in a three-dimensional cell, again, you can get very good quantitative match to their data. So we get nice stochastic oscillations, and we get the correct numbers of molecules as well. So this is very important in cancer, and we're continuing to look at that in some more detail. Okay, so on to the next scale, and hopefully we'll now use some of the intracellular modeling here as well in a few slides' time. And the next scale is to look at individual-based models of cells. So looking at a cell as an individual entity interacting with other cells, other individual entities. And the basic maths behind this is very easy. So it's nice to be in Cambridge talking about Newton's second law of acceleration. So this is basically what we're going to use in this subsequent model. So F equals ma, and then you throw away the acceleration for these cellular systems, because that's you can ignore that. And so you basically can get a system of all your forces is just some dx by dt, and if you integrate that, you'll get the position of an individual at any point in time, or a position of individuals interacting with each other in time. So the model that we've been developing over the last 6 or 7 years was originally developed by Dirk Drasdo, and it models cells as little elastic spheres, and you can allow your little elastic sphere to have a cell cycle, so you can allow it to grow to whatever maximum size your cells grow to, and then at that point you divide the cell into two daughter cells. Okay. And you, if you have more than one cell, once you have some divisions and you have interacting cells, you can introduce some cell-cell repulsion. So cells like to push each other away up to a certain point, and we do that by something called the Hertz model, which I'll show in a minute. And also cells have the ability to adhere to each other via membrane-to-membrane contacts, which are controlled by these molecules I mentioned earlier, E-cadherin and beta-catenin. I'll look into that in a bit more detail as well. So you can have cell-cell repulsion, but also cell-cell adhesion, and obviously if these forces all balance out, then you get some equilibrium configuration of your cells. So the Hertz model gives you a repulsion potential between the two cells, depending on the size of the cells and the distance between their centers, and also the elastic properties of the cells. You're considering these as elastic spheres. So this is modeling the biomechanical properties of the cell with some Poisson ratio and elastic moduli. So you can vary your biomechanical properties by changing these parameters here. And so this gives you a repulsive force, and you put some adhesive forces on, which will depend on the molecular bonds across the surface area of contact between two individual cells. And you can do that just by assuming a kind of parameter that will adhere cells together, or you can do it in a multi-scale way with a bit more sophistication. And here's a nice little movie of some epithelial cells on a petri dish. And you can see when they touch each other, there's some GFP, green fluorescent protein here. That's monitoring the levels of E-cadherin and beta-catenin across the membrane. So you can see when the cells adhere to each other, you get these molecules coming to the membrane, binding together and forming nice adhesion junctions across the two different membranes. When the cells aren't bound together, the E-cadherin and beta-catenin go back into the cytoplasm and wait until the next connection, when they're brought to the surface and they make the adhesion. So let me just speed that up. So this is what's happening inside cells when they adhere to each other. E-cadherin and beta-catenin in the cytoplasm are brought to the membrane, where they form a complex, E-cadherin beta-catenin complex. That binds to an ecoderic beta-catenin complex in the next cell across the cell membrane. And if you have enough of these complexes across membranes, you have tight adhesion junctions controlling the adhesion. And vice versa. If you pull cells apart, then these bonds break and you release the ecoderic beta-catenin back into the cytoplasm. So in each little elastic sphere, we put some ordinary differential equations because it's computationally hugely expensive if you want to do this with PDE. So we made an approximation that everything was just ODE. So this is a well-mixed reactor again, in spite of what I said in the last 20 minutes. So we have ecoderic and beta-catenin. They react together to form a complex. The complex can then be brought to the membrane when you adhere, and that can dissociate. So it's a simple set of ODEs based on the law of mass action, but this is now inside each individual little cell. So in addition to the repulsive forces, we have these adhesive forces across membranes depending on these ODEs inside the cell. So it's a multiscale model. You can calculate the force from your potential, and then you add up all the other forces in your system. So if you have your cells on some substrate, there's some cell-substrate friction, and you can put some cell-cell friction. You can add in a little bit of noise, because there's always noise in your system. These are the adhesion repulsive forces that we've calculated. You can add in some external forces, such as chemotaxis, so you can put in an external chemical that diffuses and generates essentially an external gradient, which acts as a force, or you can put in haptotaxic gradients in matrix, and again that acts as like an external force to guide the cells up the gradients. And then you simply solve these equations via Newton's second law. It's just a big system of ODEs, and you can generate the growth of masses of individual a cellular mass. So here's a little movie showing one cell. Speed this up a bit. Okay, so there we have a first division. I'll help it along. And so every 17 to 25 hours in the simulation, these cells will divide. The changing colors are denote the changing concentration levels of the ecoderic beta-catenin as they bind and unbind. And so you can see this is the development of a little cellular mass. Sometimes the cells, the daughter cells, break away a little bit, but then they're caught up by the expanding mass. And so you can use this to generate the initial growth of, in this case, it's a kind of putative cancer. Yeah.
There's no surrounding media here.
Exactly. So this is nice up to a point. And certainly if you're focusing purely on cell-cell interactions, it's fine, and you can get some information there about how cell-cell adhesion may influence the overall growth, but essentially you're growing into a vacuum, which is not very reasonable, really.
Why does it seem to be large-scale correlation in the level of the adhesive proteins? You see lightning-like things, right, going through the whole...
Okay, so that's just a color scheme denoting when you have a daughter cell born and it binds, you create these bonds across the surface. That then changes the level of, I think this is for the ecoderic level, but it's the same for beta-catenin. So that then changes the level of ecoderic in the cell, because the free ecoderic is then brought to the surface and binds into a complex. So you have a change in the concentration levels of ecoderic and beta-catenin, and that's just denoted by the color changes there.
That propagates only through diffusion?
No. So what happens is that if you take one cell and you have several cells connecting with it, then you have cell-cell interaction, and so you have all these changes in concentration levels going on in multiple cells. So there's no diffusion in the model. It's simply due to cell-cell interaction, and there's a bit of noise in the system, so it's a bit of stochastic changes, but it's just cell-cell contact. So yeah, one at the back and then two at the...
Do the single cells grow like during their cell cycle? They do not seem to be growing before division, and that might change the local force balance.
Okay, that You can explicitly model that and visually and visualize it as it grows to 5 millimeters and then divides. I think in this model, to speed things up, we just assumed that it's 5 millimeters and then it takes 17 hours to divide, and then it divides into 2 daughter cells, and then we kind of make them 5 millimeters quickly, just to help the simulation along. But you can allow them to half in size and then grow up to 5 millimeters and divide, and the overall dynamics are relatively unchanged. So, yeah. Okay.
Mine is a bit different. This is a very abstract model, and I fully understand that you need a very abstract and coarse-grained model in order to simulate on this tumor scale.
Yeah.
On the other hand, I wonder how to verify or falsify such a model, because all the assumptions you mentioned, one could question. For example, if you do AFM on a cell in the dish, the Hertz model doesn't even give you the right exponent. Adhesion is much more complicated, and so on. So, how can we basically make such models better, or find the parameters that would fit to reality?
Can you give me 2 slides?
Certainly.
Right. Okay. Good point. Yeah. No, that's right. It's very abstract. And, just to show that it does something moving you in the right direction at this point, sure. Okay, another little abstract movie simulation. Again, so this is our little cell mass. What we've done here is put on an external field of some chemotactic factor, and we've varied the strength of the cell-cell adhesion, and we've shown that essentially, if you reduce cell-cell adhesion, unsurprisingly, then the cells can break off and they can move via chemotaxis and sort of invade. So now they're invading in a vacuum. So, but again, it's a proof of principle. But in order to make this more accurate and a better model, we recognize that we would really need to have some interaction with matrix in some sense. So what we did was go right back to a single cell and allow the single cell to interact with some matrix fibers, because there were some experiments that we'd seen in the literature of people putting single cells or a small number of cells on some collagen with varying levels of collagen in the matrigel. So we took our little cell, we're going to simulate in a 2D domain, so it's a hemisphere now when it's on our substrate, and we interact it with some fibers which we're assuming are just rigid rods about 75 millimeters in length, 200 nanometers wide, and they're randomly distributed on our little square mathematical Petri dish. Okay, so we're wanting to look at things like cell migration, cell polarization, matrix fiber remodeling in terms of interaction with a cell. And again, we did that in a simple way. We assumed that the individual cell could just simply pull on the matrix fibers just using a force and turn it through some angle. And if it interacted with several fibers, it did this to the several fibers, and then if you kind of do a weighted average of the fibers and the strengths, that you got some good data from AFM experiments of the forces that cells put on fibers. You can use interactions with multiple fibers to essentially allow the cells to crawl along these fibers. Okay, so I won't go into the details too much with the lack of time, but I'm happy to talk about it afterwards. It's all been published. So now what we have is our little elastic cell, which we can grow and divide and have all these things, but we're now focusing on interaction with matrix fibers. And we looked at some work from Mike Adembo's group in Boston from about 15 years ago, and they'd put a single cell in matrigel, and they'd made half of the matrigel soft and the other half stiff. That was all they said. They didn't really say what that meant, so we had to guess what that was. So we made a little mathematical Petri dish. Half of our matrigel was soft, so it had lower density of fibers per unit area, and the other half was stiff, had a higher density, and they were harder to move. And then what we did, we just ran the simulation looking at how a cell moved over time by interacting with the fibers and moving along the fibers. And see, because they had not really quantified what they meant by soft or stiff, the initial simulations that we did were rather, well, they didn't really say very much. Sometimes they moved to the left, sometimes they moved to the right. But when we really made a difference between the softnesses on the two halves, what we found was that if you placed a cell at the border, then on average, the cell would like to move from the soft side into the stiff side, or vice versa. If you placed it on the stiff side originally, it preferred to stay there. And that was exactly what Dembo had found in his experiments. That if you put a cell on the soft side close to the boundary, then on average it would go to the stiff side, and vice versa. And they had a complicated explanation for this in terms of signaling molecules and all sorts of things that were going on, whereas our simple model here simply seemed to indicate it's a biomechanical problem with interaction with the fibers. Yeah.
Cells were you using?
So our cell was just this little elastic hemisphere with some elastic parameters, which we tried to. But these guys were using fibroblasts, I believe.
I don't understand how you simulate the interaction between fibers to obtain different elastic modulus.
The fibers are all just rigid cylinders.
Yes, but how they can interact or not?
The fibers don't interact with each other, but if a cell interacts with several fibers, it pulls on all of the fibers individually, and then you do a weighted average of how the cell has moved each fiber, and then you move in a direction that's kind of averaged over the directions of the different fibers.
Where comes the stiffness into play?
The stiffness is we can well, it depends. It's the force that the cell has to impose on the fiber to move it through a given angle. Okay. So if you have a stiff matrix, then it's harder for the cells to exert a bigger force to pull the fiber through a given angle.
So you haven't put contact guidance in yet?
No. No. So this is just, as I say, purely mechanical. Okay, so again, if you look into this in a bit more detail, then, and this has all been published recently. Okay.
There's a stick somewhere.
Good.
Yeah, it's on the other side of the board.
Excellent.
Yeah.
Yeah. So you can look at what happens by changing fiber length, fiber density, the stiffness, the direction of the fibers. All of those have an influence on how fast the cell will move through these matrices, and the persistence time, i.e. how long it will go in a given direction before it changes. So, and again, you can map that onto experimental data in this area, and at least qualitatively you can...
I think it's gone.
Okay. The battery's going to... Okay.
Yeah. This is, yeah. No.
I'm needing a new Duracell battery in me, I think. Yeah. So anyway, right, back to manual everything now. Okay, old school. So yeah, so the matrix stiffness in particular has an influence on the speed. And you can, of course, align the fibers and make it more anisotropic or less anisotropic, and that also plays a role. And you get some interesting non-linear relationships in terms of the persistence times of the cell movement and the length and the density of the fibers. So that's all tries to be a bit more quantitative and show that this modeling approach isn't just nice. You can make nice movies, but does map onto real data that people have measured. And then you can move things up. It just takes a little longer to compute. Here are two cells interacting, and by changing the fiber alignment, you can get cells kind of playing follow my leader. So we have a lead cell here that we've polarized, and initially it moves in a given direction, and this cell behind it isn't polarized, but because of the fiber alignment caused by the lead cell, the trailing cell becomes polarized as well, and as I say, it plays follow my leader for a bit, only down to the interaction of the lead cell, and then you can simulate, tens of cells in this little two-dimensional fiber, and you can see fiber alignments, and we're trying to explore this in terms of cancer cell invasion by interactions with the fibers and degradation of the fibers.
So this is a system of OD coupled the mechanics model.
Yes. Yes. This we don't have any. We don't have any cell E-cadherin beta-catenin in this particular model because it's mainly interactions with the fibers we're interested in. If two cells interact, we just have some adhesion value that they stick to each other. It's not controlled by these. Obviously, we could put that in, and that would just make the simulation far longer because it's computationally quite expensive doing all these little forces and interacting with the fibers. Yeah. Okay, so the other thing that we looked at is use this model to try and get an insight into the metastatic process, focusing on the interaction with a cell, a cancer cell in red, and a blood vessel. Now again, there was some data showing that there was a protein called SARC, and if SARC is mutated, then the cancer cell exerts a greater force, and people have looked at this in some detail and measured the time that it takes a cancer cell to penetrate a blood vessel with SARC mutated or SARC as a wild type. So this is just the simulation of these trying to capture these data. So what we have here is a cancer cell, and there's a cylindrical tube of green endothelial cells, which is a very basic blood vessel, and they're all connected together with our E-cadherin adhesion system, so they're at steady state. So the cancer cell is going to move towards that with some external force at a given rate. So this is moving with a speed that you match with data. And so when it gets to the blood vessel, it's then going to bind to the endothelial cell and start disrupting the E-cadherin beta-catenin bonds. So here's the cancer cell. So you can maybe see here now that the green cells have changed color and they've gone a bit yellow, so that's disruption of the cell-cell bonds. That's releasing the E-cadherin beta-catenin back into the cytoplasm and reducing the cell-cell contacts. And we simulate this for an appropriate period of time in terms of experiment. And in this case, what's happening is that the cancer cell has exerted sufficient force in order to squeeze between the endothelial cells. So that's done with the parameter values. So this is estimating the force and the Sharon.
How is it disrupting those bonds? Just pushing, or is there some ligation?
Well, when it moves with a given speed and hits the cell, it will exert a certain force, and that will allow a certain overlap in cell area between the cancer cell and the endothelial cells. Okay, then you can estimate the number of bonds that are in a given area, and so you disrupt those bonds with that force, and so that then releases the E-cadherin and beta-catenin back into the cytoplasm. Sorry?
Mechanical.
Just purely mechanical, exactly, yeah. So, and this maps very well onto the data. It takes about 30 minutes to do this process, and that maps back to what the people have measured. So this is, again, trying to get a bit more quantitative to estimate what you need, and it's the right force, the right time, and it's managed to succeed. So you can try and develop ways to stop the cell moving into the blood vessel, because clearly, if cancer cells don't get into blood vessels, then they wouldn't spread and you wouldn't get metastasis. So if you can focus on mechanical ways to stop this intravasation process, then I think you would go a long way to stop metastatic spread.
How do you model your blood vessel?
Oh, this blood vessel is just, I mean, this is very, very, this is a very, very naive blood vessel in the sense that it's just a cylinder of cells all stuck together with these adhesive forces.
How do you estimate these adhesive forces?
Sorry?
How do you estimate these adhesive forces?
Well, so people have taken cells and they've pulled them apart, and then they know the force that it takes to pull the cells apart, so you know what force is holding the cells together. You can estimate the total number of ecodherm receptors on a cell surface, let's just say 10,000. So if you have a quarter of the cell surface area, you know you have a quarter of 10,000, and so that will give you a certain force on that surface area of overlap. So the calculations are pretty straightforward, and there's a lot of data there that you can use to parameterize your model.
How important is it that the cell probably deforms itself enormously as it squeezes through?
I think that is important, yeah. And I think, well...
How would it change your estimates? Would it be easier, I suppose?
Okay, so I think, so we assumed here that if the cell exerted a sufficient force and broke a sufficient number of bonds in a given time, it would get through.
So you don't really worry about it squeezing them?
No, I mean...
it just sort of gets through.
It gets through, exactly. I mean, Luigi's given a talk here. Did you talk about your nice cell with the hard nucleus? Okay, so Luigi's done some really nice work.
I didn't do the encapsulation through the material layer.
Yeah. So, I mean, Luigi's got a lovely model where cells got a stiff nucleus or a soft nucleus, and he can model cell shape change and penetration into small or large channels. So that is definitely happening, and a more accurate model should take that into account. But we've simply looked at the kind of measured numbers here, and the cell exerts an appropriate force, breaks an appropriate number of bonds in an appropriate time, and so then you can say it's successful.
The actual structure of the blood vessel in the cell itself isn't terribly important here. You're just looking at the forces, number of bonds.
Yeah, exactly. This is a very schematic movie. Yeah, sure. Okay. Yeah.
So you said that you get the forces from pulling the cells apart, but this is more of a pushing. So it's sort of a slight difference. Do you think there's any mechanical differences in the interactions because cells might respond differently to a pushing force versus a pulling force? Or do you not want to think about that? Because I wouldn't, but
It's not that I don't want to think about it, but if it becomes hard when I think about it, I just ignore it and just say, well, we'll just use the pulling force.
Just at least acknowledging that there might be a difference, or?
Oh, sure. No. A lot of this work was done some years ago, so things may have changed, and there might be more data out there now or more experiments done. At the time, we used the available data that we could find, which was from AFM experiments on pulling cells apart. So we made the assumption that wasn't such a bad estimate, and that's what we used. So if it's wrong, we'll have to go back and do the whole thing over again with appropriate new data. Okay. So 10:15, is that okay? Yeah, okay, all right, I should speed up. Yeah, you see how many slides I've still got left anyway. I'll speed up. Okay. So this shouldn't take too long because it's a PDE model and not so much to explain. Yeah, so this is now going to a tissue scale. Well, you know, I'm sure you all know about PDEs. This is a reaction diffusion. It's all back to mother pie and nice comfortable things. So this is the invasion process looked at a tissue scale with PDEs. And so we're going to focus on three key aspects of invasion. The cancer cells themselves, they secrete various enzymes. So we're just going to consider a generic enzyme, a matrix degrading enzyme, and then that degrades the extracellular matrix V. So secretion of enzymes, degradation of the extracellular matrix, and then the cancer cells interact with that degraded matrix, and we'll see what happens. So you can write down a system of PDEs, as I'm sure a lot of us do. Here are the cancer cells. There's a bit of diffusion and proliferation with taking into account the competition for space with the ECM. The ECM is degraded by MDEs, and it tries to get back to its normal state. So there's some kind of sort of restoring term here. And the MDEs are produced by the cancer cell, and they diffuse and decay. And you can, maybe 15, 20 years ago, for this component here, you would write some haptotaxis function. Okay, so when the matrix is degraded, you get changes in the matrix density, which leads to a haptotactic response from the cancer cells, so they prefer to go into regions of higher density. So if you do that, you've got a nice system of PDEs with a kind of logistic term here, which is not a million miles away from Fisher's equation, and so you get nice traveling waves. Fine. And you can estimate the rate of invasion and how quickly it invades and all those kind of things, as you do with traveling waves type solutions. But that's it. Once you start with some initial data, you're just going to get invasion. Or to not get invasion, you have to do something drastic, like stop the cancer cells diffusing, stop them proliferating, and essentially, you don't have cancer cells. So we'd realized that, and although we'd played about with these reaction diffusion traveling wave type systems, we were trying to make it more accurate and have some better way to model the interaction with the matrix by the cancer cells in a PDE model. So some years ago now, Alf and I wrote a paper where we borrowed some ideas from Jonathan Sherritt and his group, who looked at cell sorting, modeling cell sorting experiments. So put two cell types in a petri dish, and cell type A and cell type B preferentially adhere to each other, and they split apart. In fact, that was probably the basis for CompuCell 3D all those years ago, James, differential adhesion and sorting. So in order to do this with a PDE, we used the idea from Jonathan Sherritt. I'll just go through this very quickly, which is basically to introduce a non-local term. So you have a cell, and it's got some little epsilon ball around it that it interacts with other cells and matrix. And I'm just going to run this through this very quickly. So you have now a non-local interaction term between the cancer cells and the matrix, but you can also have a non-local term between the cancer cells and themselves. And because it's non-local, you don't get complications with backward heat equations and ill-posed problems from a PDE point of view. Okay, so it's a nice way to model it. And you can have a cell-cell adhesion coefficient and a cell matrix adhesion coefficient. And if you do the maths and let this little radius of influence tend to 0, you recover the standard haptotaxis terms that you would expect if you just wrote down the PDE without going through all this trouble. And you recover, if you look at the cell-cell interaction, a kind of singular term that leads to a backward heat equation, which is why PDEs like this don't work with kind of self-haptotaxis of cancer cells. So it's a nice way to model adhesion using PDEs. And we've looked at these systems, and we've added in a bit of mutation in the system. So for example, we take one cancer cell type, and we allow it to mutate, and it becomes a second cancer cell type. And the second cancer cells types have some different parameters. They will proliferate more, they'll have less adhesion. So simple changes into the model. And we'll just then...
compete? Do the different clones compete? For food or oxygen or something?
So no, we don't really have any notion of oxygen or anything like that.
No competition between the different ranks of cancer cells?
Well, there's competition for space.
Ah, okay.
Okay, here in this sort of modified logistic term here, yeah. Okay, so now we did this, Alf and I did this a long time ago with constant adhesion parameters, these SCC and SCV, and recently we've updated the model and we've looked at time-dependent adhesion parameters. So what we did was assume that as cancer progressed and it got more aggressive, the cell-cell adhesion, as is known, cell-cell adhesion reduces, so cancer cells become less adhesive to themselves and they become more adhesive to the matrix. So you can have a kind of monotonic decreasing function for SCC over time and a monotonic increasing function for SCV over time. And when we simulated this, we got some really nice results from a purely aesthetic point of view initially. So we just got some cool patterns evolving rather than just expanding into the tissue. And obviously if you change your parameters with these adhesions, you can stop the cancer invading if you have strong enough adhesion. But with these time-dependent parameters, we got some very interesting patterns evolving.
5 minutes.
5 minutes, thanks John. I'll finish in 5 minutes. So you change the parameters and you get some interesting patterns evolving, as I say, rather surprisingly. This is for a two population model, so you get kind of heterogeneity evolving quite naturally. And then we thought we should try and look back to the data, and in fact, these heterogeneous invasion patterns actually are what is observed. These are two. This is a ductal carcinoma in situ, and this is another phyllodes tumor. I'd have to look that up myself what that is now, but anyway, you can see you get the dark stained cells are the cancer cells embedded within the matrix. And there's been some recent work by a Japanese group trying to classify invasive patterns in general, and they've come up with this infiltrative growth pattern classification. This is an infiltrative growth pattern A, which is a good situation. These are the cancer cells here. This is the matrix, so there's a nice demarcation between the two. This is infiltration pattern C. This is a very heterogeneous mixing of cancer cells and normal tissue. And then this is infiltration pattern B, which they simply say it's somewhere between the two. So this is good, this is very bad, and this is somewhere in between. Yep.
Is that the same tissue treated with different things or different tissue?
No, this is the same tumor, just in a different stage of malignancy. So this is less malignant and this is more malignant. I'd have to look. Yes, so there it's esophageal cancers. Okay, and then we've also tried to model some in vitro invasive assays. These are called organotypic assays. This is a collagen gel and some cancer cells on the top. And if you change a component in the matrix, if you play about with the collagen, you can get a more heterogeneous invasive pattern. So just very quickly, we can do that in our model. Normal invasion, change cell matrix adhesion parameter, and we get a more heterogeneous invasion. So again, this type of model is trying to map back. We're trying to map this model back onto real data and make more accurate predictions with the parameters in our model. Okay, so just very quickly, some future perspectives. Well, hopefully in the years to come we can try and put all this together. This is the intracellular stuff, cell-cell stuff, and the PDE model with some maybe high-powered computing to bring this up to tissue scale. And I haven't talked at all about angiogenesis, but hopefully we're moving towards trying to get some virtual tumor, whatever that might mean. And just before I finish, I'd like to thank my collaborators on this work, Mark Sturrock and Maria Potashnik, who worked with me on the intracellular HES1 P53 work, Ignacio Ramis-Conde and Daniela Schluter, who did the work on the individual cell-based model, and Alf, whom you've heard talk earlier here, Pia Domschke from Darmstadt, and Dimitri Trucu, who've worked on the PDE model of invasion with cell adhesion. So I'll leave it there, and thank you for your attention.
Magnificent, thank you, Mark. Any further questions before tea? Okay, people keen to get their coffee.
Yeah.
To talk to Mark
Yeah,
over there.
please do. Yeah.
Reconvene in half hour.
Thank you.
Because he was flying over.